Hello, good afternoon. Hello, good afternoon. Thank you all for joining us this afternoon in sunny Vienna. So I hope you can all still stay a little bit here with us until you are free and available to go out and enjoy the sun here in Vienna. But today we have still a very interesting satellite symposium to show to you. But just let me start first with introducing very shortly Medicines for Europe. Medicines for Europe is formerly known as EGA, or the European Generic Medicine Association. We just recently rebranded for the reason that we represent the pharmaceutical companies that supply the largest share of medicines across Europe. Our members have in Europe about 350 manufacturing sites and employ around 160,000 people. What a lot of people do not know is that we also invest in R&D, so 70% of our turnover goes back into R&D of generic, biosimilar, and value-added medicines. And the medicines that are supplied by our members cover 80% of the therapy areas. Our association has a vision. This vision is built around 5 key pillars. The most important pillar is patients. Everything is built around patients with the aim to provide high-quality medicines to patients. The value that our members bring is not only in savings, cost savings, but also, for example, increased access to medicines. We also ensure sustainability for healthcare systems, and our aim as well is to create a sustainable industry as well. And we work a lot with our partners, like, for example, today, EAHP and the hospital pharmacists. Now, to give you a brief introduction about the 3 sector groups, as I mentioned, we have generic medicines group, we have a biosimilar medicines group, and we have a value-added medicines group. Thanks to generic medicines, over the past 10 years, twice as many patients have been able to be treated in 7 key therapeutical areas. Generic medicines take into account 56% of the medicines that are dispensed, while only taking up 22% of the pharmaceutical budget. If you do a recalculation, this is only 2 to 4% of the healthcare budget. Biosimilar medicines, that's why we're here today. Our members have 10 years of experience of biosimilar medicines on the market, and 20 products have been successfully launched in that period. The aim with biosimilar medicines is to bring more competition to the market in a market of biological medicines, which is a high-value market. We also have recently started up a new sector group, which is called the Value-Added Medicines Group. This is actually a group which lies a little bit between generic medicines and innovative medicines. So by redesigning and redeveloping already known molecules and improving them, we improve an added value which can go either to patients, payers, or physicians. This is only a teaser because this group will only be launched in May 31st in Brussels, and you're all welcome. But today we're here to talk about biosimilar medicines. I know you probably know very well the speakers that are here today. Some of them also presented already yesterday in one of the sessions, but they wanted to tell me that yesterday was the course for beginners and today is the advanced course. So let me introduce you in order. First of all, Dr. Niklas Ekman, who is a senior researcher of the Finnish Medicines Agency, and he is also a member of the Biosimilars Working Party, and he will give you an update from the regulator perspective on biosimilar medicines. Our second presentation will be from Professor Arnold Vulto, who is a professor of hospital pharmacy at the University of Rotterdam. And our third distinguished speaker is Dr. Korns, oncologist, who is a big fighter for biosimilars. So I would like to welcome for a first presentation for Dr. Ekman.
So, thank you very much for the introduction. Yes. So, what I will do is to give you a short overview of the regulatory approach for biosimilars that we have in Europe. Is it okay to a little bit? No, I can see it. Thanks. So, as said, first of all, I'm going to go through a little bit, give some details also about the principles, how they are developed, and especially how we regulate biosimilars in Europe. After this, I show you something about the current biosimilars as well as what can be anticipated for the future. The last part, or the last few slides I have, just shortly again gonna tell about our position on a hot thing related to the interchangeability of these drugs. So, as you know, biological medicinal products and biotech products are produced or produced in living organisms. So for biotech products, for example, we are talking about cells, which of course then also means that there's a lot of variability always built into the system and manufacturing systems. So no matter whether it's an original medicinal product or a biotech biosimilar, no two batches will ever be fully identical with each other. Another thing also which plays in here is that manufacturing changes happens a lot both for originators and for biosimilars. You can see here on the left side of the slides from a publication from Christian Schneider, where they actually looked into these manufacturing changes. And for example, Remicade, so infliximab, you can see that over 35 manufacturing process changes had taken place already in 2013 or 2012. This, of course, doesn't mean that all these changes are or will change the quality profile of the products. However, some of these will have some impact also on that. So in order for the company or manufacturer to introduce these changes into the manufacturing process, the regulators require that the companies are testing the pre-version and the post-version of the drug in order to show that both the efficacy and the safety is the same before and after the change. This is something that we as regulators and the companies have been doing already for like 20 years or something. So it's not a new thing to assess for comparability. So if we then go to biosimilars, so what are actually biosimilars? Well, I think there are two important messages in the current EU regulatory definition of biosimilars. First of all, it says that biosimilars contains a version of the active substance. And it's a version of the same active substance as the reference product. So in other words, it's not a new active substance. It's the same active substance, just a different version of that one. So very much similar to the manufacturing change issue that I just described. You have a pre-version and a post-version after the manufacturing process change. The second important thing here is in the definition that it also says that you need a comprehensive comparability exercise consisting of all different levels, the quality characteristics, biological activity, safety, efficacy, and study these in order to show that there's really no difference. The scientific principle, as I said, for biosimilars, or the comparability exercise for biosimilars, are actually based on the very same principles as used for assessing manufacturing process changes. So in principle, the concepts of biosimilars is applicable to any biological medicinal product. However, in practice, there are, of course, some things that need to be taken into account. First of all, you need to be able to manufacture a very close copy version of the originator. Secondly, you need to be able to perform thorough physicochemical and biological characterization. as well as also to understand the clinical relevance of any differences that you encounter. You need to be able to demonstrate bioequivalence in pharmacokinetic studies, also pharmacodynamic studies if possible. And there also need to be suitable clinical models with sensitive endpoints and a possibility to identify relevant comparability margins. So on a very high level, then how close is close enough when we talk about biosimilars and reference? Well, the amino acid sequence as well as the dosing and the route of administration need to be the same for the biosimilar and the reference product. Then the active substance, as I said, there are always some small differences between different batches, for example, different products also. So the active substance in terms of molecular and biological characteristics need to be similar or highly similar. Highly similar is actually something that FDA use in US. However, in Europe, both the legislation and guidance and regulatory language just use the similar. If there are differences in strength, pharmaceutical form, formulation, excipients, or presentation, these need to be justified. However, it is possible to have those. What is not allowed is to make intended changes to improve efficacy. So when the biosimilar companies start to develop the biosimilar, there's a lot of different things that they need to keep in mind. For example, here now, this is a slide on a monoclonal antibodies. There's a lot of different, both quality attributes, but also then physicochemical characteristics as well as biological and functional characteristics that need to be taken into account. Here I have just summarized a few of them. There are hundreds of different characteristics that really need to be shown to be similar between the two molecules. The good thing is, however, that there's also a lot of highly sensitive and sophisticated analytical tools to characterize these molecules. The second thing is also that there has been a tremendous development in the sensitivity of these tools in late years. Just to take one example, for example, mass spectrometry, which is very much used for different purposes. In the last 10 years, the sensitivity has gone up approximately approximately 10,000-fold. So it's really, the message is here that first of all, we have a lot of different tools to study one and the same thing. And in addition, the tools are very sensitive. So on an analytical level, we really can go into the molecule and see what the possible differences are. Of course, when the biosimilar company developed the biosimilar, the whole aim with the development is to get as close as possible with the originator. After the developmental work, so when the final commercial manufacturing process is established, the company needs to do the so-called pivotal evidence for analytical similarity. So this is very extensive. It's a side-by-side, whenever possible, comparability exercise to show that the two molecules are similar. Composition, physical properties, primary high-order structures, purity, product-related isoforms, impurities, biological activities, and so on. So all kinds of characteristics need to be considered. The company always needs to use orthogonal methods in order to see from different angles at the same characteristics that whether they are same or not in the biosimilar and the originator. For this pivotal analytical similarity exercise, quantitative comparability ranges must be established. These ranges are based on the data that the company has on the reference product. So the characterization data using those exact methods that I showed you in the previous slide. Differences are always seen. As I said, the analytical methods are highly sensitive, and we have a lot of different of those that can be used. However, any differences that can be seen between the biosimilar and the reference product must be justified with relation to the possible effect that it could have on safety and efficacy of the molecule. It's also clear that clinical data cannot be used to justify substantial differences between the two molecules. If there are differences, or when there are differences, it of course also matters what kind of quality attributes we are talking about. So if it's a low quality or a low criticality attribute, usually it's enough with previous knowledge to justify the differences. However, very often also, I would say it's more a rule than an exception. The company needs to do real extensive more work in order to understand what the differences could be or mean in the clinics. So actually when the biosimilarity is established between the biosimilar and the reference product, one could think about 4 different levels where it's established. It starts with the comprehensive chemical and biological characteristics. After that, non-clinical studies are conducted. In vitro functional studies are really the paramount studies. At the moment now, in vivo studies, comparability studies are usually not conducted in Europe because our experience has shown that with regard to comparability, there actually is not very much that can be gained from those studies. The third level would be the pharmacokinetics and pharmacodynamic studies. They should be done or the PK study should be done in a sensitive, homogeneous study population, usually healthy volunteers, if that's possible. Then the final level, so the confirmatory level, is the efficacy and safety study. It should be powered, adequately powered, randomized, usually parallel group, however also crossover studies have been used. And they are usually also equivalence trials. The study population must be representative of approved indication, but more importantly, it needs to be sensitive for detecting potential differences, because the efficacy of the molecule, of course, does not have to be established again. That has been already done for the originator product. The same goes with the endpoints, and just to say that also safety data, comparative safety data between the biosimilar and the reference product is always needed. Usually the efficacy and safety data is requested from one sensitive indication in order to support authorization of all the indications approved for the reference product. So extrapolation of indication is what is done. However, this is not an automatic thing like for generics. When we're talking about biosimilars, always it's a case-by-case situation where the totality of evidence brought up by the company is considered. And very importantly also, the scientific justification provided. So to summarize, on the left hand, you can see the situation with the originator product, and on the right side with the biosimilar. As you can see with the originator, the clinical studies actually are a huge part of the package. For the biosimilars, the pyramid is a little bit different. The main part of the studies are at the analytical levels, because those are the most sensitive to detect differences between the biosimilar and the reference. However, comparative in vitro biological characterization is also done, as well as, at the moment at least, still always also some kind of comparative clinical studies. So using this scheme, we so far have assessed 30 different biosimilars. These have ended up in 21 positive, 7 withdrawn during the evaluation, as well as 2 negative positions by the CHMP, so the Committee for Human Medicinal Product. Currently, 19 biosimilars hold a valid marketing authorization. You can see them listed here. And 2 authorization has been withdrawn by the marketing authorization post-approval. This field is actually very, very active. At the moment now, there are 8 new biosimilars undergoing evaluation or assessment by the CHMP. And interesting enough, actually 5 of these are totally new active substance. So we have adalimumab, enoxaparin, pegfilgrastim, rituximab, and a teriparatide that is currently undergoing evaluation. For the future, what I can say is that the biosimilars are going to increase. This is from scientific advices given by the CHMP the last 10 years. You can see it's a trend upwards. And the second thing also that can be seen from the the red columns here is that the follow-up advices are increasing all the time. That means that the advices that we are nowadays giving are more towards more mature products. So it's in the upcoming years, we're really going to see a lot of these. At the moment now, some 70 to 80% of the scientific advices concerns monoclonal antibodies, so that's obviously going to be the biggest part. Okay, then for I think it was Tri's last slide, I just want to inform you about a position paper that we have published from the Finnish Medicine Agency on the interchangeability of biosimilars. Because when EMA and CHMP is evaluating biosimilars, we are just evaluating for marketing authorization purpose. So the interchangeability, as you for sure know, it's a national issue not covered by the EMA evaluation. Just to give a little bit of background, the funding of medicines in Finland, how it works, those medicines that are administered in public hospitals are covered by the communities, while the medicines delivered at pharmacies are reimbursed by the state. And of course, for biological medicinal product being so expensive as they are, reimbursement is a prerequisite for the use of these drugs. We have 3 different levels of reimbursement, and the biosimilar automatically enjoys the same level of reimbursement as the originator. Currently, hospitals are the leading in the adoption of the biosimilars, while we still have some issues and problems with getting really prescribers and patients on treatment to get involved in the biosimilars. This is clearly due to the fact that there's no incentives for those groups. So as I said, this paper, it's published on the internet. It's very difficult to find it. That's why I put the link here. But it defines the current position of our agency towards interchangeability of EU biosimilars and their reference products. It's also a recommendation for our healthcare system. The position paper really talks about interchangeability, not about automatic substitution, which would take place at the pharmacy level. The main conclusion of the position paper is the following. Switches between biological products, they do happen in the clinics all the time. For example, EPOs have been changed or switched for long times already, and usually there's not any problems encountered with this. We also have clinical evidence from crossover studies conducted, and so far at least no evidence of adverse effects due to the switch from one good product or high quality product to another high quality product of the same active substance has been encountered. I won't go into this, but actually also the theoretical risk for adverse effect due to the switch is very weak. And what we think is that the risk of adverse effects can be expected to be similar as the risk associated with changes in the manufacturing process of any biological products. So our position is that our EU biosimilars are interchangeable with the reference product under the supervision of healthcare persons. Of course, the switch should be documented, but this goes the same no matter whether we are talking about biosimilars or originator products. Also, other national authorities in Europe has come to the same conclusion. You have in Netherlands almost the same. Paul Ehrlich Institute in Germany has a position paper saying more or less the same, as well as also the Irish Health Authority. The links can be found down there if you are interested in more details about those. With this, I would like to end, and thank you for your attention.
Thank you, Dr. Ekman. If you are okay, we will keep the questions until the end of the three presentations, because the presentations are quite complementary. So it's possible that you have a question now, which will be answered already by one of the next presentations. So that's also why the presenters asked me to wait until the end. So next presentation will be from Professor Vulto.